So we saw a slide like this something before about the drug discovery productivity and literature reliability. And I think the statistics on this one are the worst you've chosen. This comes from various papers you would discuss. But the central problem is, if we're a pharmaceutical company who wants to do an innovative drug discovery program, what information do you need to be at the starting line? So if you look at how compounds progress from preclinical through to registration, the big fallout is in the phase 2 trial. And this is where the hope for hypothesis runs into human reality. And this is where only 25% of compounds, I think this is for cancer, go through the phase 2-3 transition. So some of this is down to compound quality, but at least half of the problem is down to the poor choice of underlying hypothesis. So target validation is a problem. So one of the reasons for this is that these programs have been triggered by, in the old days, internal research, but more recently academic research. And here's this famous paper from Bayer, who were quoted in Nature Reviews Drug Discovery. And when they tried to replicate results of 67 studies in Cell, Nature, and Science, nearly 2/3 failed. Perhaps not completely, but the key piece of information which would give you confidence to move forward didn't come up. There's some more data at the bottom from Amgen. 6 out of 53 were the only one, a small fraction reproducible. 14 out of 67 for Bayer. So this goes into de-risking strategies. So one more thing you can do is form a consortia, like the one in Dundee. So you have a number of farmers, and they're funding and providing some expertise to a recognized center, a really specialized center, and leveraging the expertise of that. So this gives these pharma companies both expertise, but also accelerated access to information. And therefore, they've got a sort of both an informational advantage and also a time advantage over the competition to build their novel drug discovery programs going on. But because the consortium members all have access to the same info, they all have the same n-month lead, which they need to act on, and on the assumption they're all full of rational decision makers, they may all pick the same targets to go forward. So does this sort of model predict portfolios will be similar? So there's a bit of straw man arguments going on here. But if you look at the drug discovery activity on kinases, it is really just concentrated on a few targets. This is data from Stefan Knapp's paper in Nature Chemical Biology in 2010. But clearly, an organization like the Dundee has got the bandwidth to look at other targets and will be trying to persuade, and influence the pharmaceutical members of the consortium to move beyond the obvious and go on. But clearly, we do have a problem with portfolio concentration in kinase research. We have an arguably unnecessary 39 compounds and antibodies against VEGFR in the clinic by 2010. So, but this model is very successful, but it doesn't suit every purpose. So Horizon does many things, but one of the things we have is a target ID and validation business. And what we can bring to the party here is cutting-edge expertise in gene editing. And we also built a synthetic lethal screening platform under advice from Chris Lord and Alan Ashworth at the ICR. And we have a belief in personalized medicine that if we hit the right target, you will hit the right response. So we found a collaborator over in Boston who felt much the same way. This is H3 Biomedicine, which is a sort of biotech company set up by Golub and Schreiber at the Broad Institute, wholly owned by ICEYE. And they're based on bioinformatics and diversity-orientated synthesis chemistry from Schreiber. So one thing that is a key differentiator of H3 Biomedicine is that they're very, very confident in their ability to do novel chemistry. And so they're unafraid to take on the major drug discovery challenges of working with first-in-class targets. And they also believe that hitting the right target will leave the right response. And so what they wanted to do was provide obtain additional validation information which wasn't in the literature, which would let them put the effort into these high-risk, technically programs to do drug discovery without getting early feedback from chemistry that they're on the right state. So they wanted very detailed biological validation. So the space where both Horizon and H3 are operating is synthetic lethality. So here is, this is from Tamburello's, one of the TGCA papers. And, excuse me. So what we have here on the x-axis is the proportion of cancers which bear a particular mutation. The most important ones have got a different y-axis from the other ones here. And so you see p53 is standing out about 35% of cancers. And then there's a long tail of falling away. But if you look in green, green are the things which are conventional, actionable, dominantly active oncogenes, like PI3 kinase and EGFR. So these are things which can be and are kinase inhibitors to deal with. But most of the problem with cancer-driving mutations is that they're either undruggable oncogenes, like KRAS, at least was thought to be until recently, or they're missing tumor suppressors. So one has to come up with a different approach to deal with this. And this is one that's been exemplified recently by Kudos and later AstraZeneca's work on olaparib, Lynparza. And so we formed a couple of alliances with H3 Biomedicine. So the first one was a target validation alliance. And this was a 4-year project where Horizon, in concert with H3, performed this very detailed validation on 5 targets, which are either coming from the literature or from bioinformatic analysis. And into this sort of consortium, H3 put cash, because we didn't have any at the time, a bioinformatics resource, some FTE resource, and the hypothesis to validate. Horizon were providing FTEs at a discount rate and the cell line engineering IP. There was also, and in terms of what comes out of this alliance, validated, or also equally importantly, devalidated targets go back to H3, and Horizon's rewarded for the discount provided on the services by a success-based milestone cascade. And so the idea is that both H3 and Horizon get a bit bigger. There's also a second collaboration called Synthetex, which is a multi-platform synthetic lethal target ID play. And again, H3 are putting in cache bioinformatics, and in this case, contributing an shRNA screening platform. And Horizon were again providing the manpower, cell line engineering IP, and an siRNA screening platform, pretty much intellectually descended from the one that had been established at the ICR by Chris Lord and Alan Ashworth. And again, in this case, both parties got access to exclusive targets. So how do we handle this? So in Synthetex, the initial target picking mechanism was a complicated round-robin basis with a sort of joker, which could be called by companies selecting a particular favored genotype. We didn't find this work very well, and also neither party at this pre-competitive stage, so to speak, had an incentive to validate the target unless it was going to end up in our pot, so to speak. The second issue was that a third party, an additional partner, was needed to run this program on the scale to have a more likely probability of delivering top quality targets. And it was difficult to identify one who had both an interest in the concept and a fit for the business model we'd already established with H3. But eventually we reconfigured the deal to get rid of some of these problems, and so that each party had an exclusive genotype from which to pick from. And this enabled Horizon to form a second collaboration with AstraZeneca to take a couple of these genotypes through, which is still going on. In the second collaboration, the Target Validation Alliance, in this case, Horizon's milestones are linked to project progression through into the clinic, and therefore there's the value we add to H3 by telling them these projects are completely hopeless isn't recognized. And there's also been some timing issues. We're impatient to get on with the work. We've got manpower dedicated to this. But H3 obviously only have a certain capability to take on targets at any particular time. So I'm going to briefly, in the last few minutes, discuss some of the work we've done in the Target Validation Alliance. So this is the first target we worked on. And this, there was a paper in Cancer Cell and also Cancer Discovery from Barbacid in Madrid and Tuvacin, who was in Cambridge at the time, both using mouse models where an activated allele of KRAS causes lung cancer. And then they were running this sort of tumor initiation model on the background of various genetic changes. For example, Barbacid showed that in a BRAF double knockout mice, the KRAS allele induced lung cancer with unaltered kinetics. But in the CRAF deletion, there was a block on cancer formation. And these other mice here, they still succumbed to cancer because they hadn't properly deleted the CRAF. In this case, the KRAS initiation and the CRAF deletion happened simultaneously on a cell-by-cell level. So that was the hypothesis. And so we did a pretty ambitious program of work to check this out using shRNA. So it's 8 KRAS wild-type cells, 13 KRAS mutant cells, multiple shRNAs against both KRAS and CRAF, plus controls. And so a typical result in a KRAS wild-type line was, much as we expected, that CRAF deletion or CRAF depletion, I should say, was relatively well tolerated. We got good levels of knockdown by RT-PCR and Western blot, and little effect on growth. In the KRAS wild-type cells, some of our best results were in A549s, but here we found that although all 3 shRNAs gave a good depletion of CRAF, only 2 had an effect on proliferation. So do you go with the weight of evidence, or do you say, well, this is a problem? Here we are, we're knocking down CRAF, and we're not getting an effect. And so this isn't directly in contradiction to the mouse results, because they're about tumor initiation rather than the maintenance. So we've moved on from shRNA to CRISPR. Again, got ambiguous results, but now we've been able to, using Horizons technology, characterize multiple clones of A549 cells, where we've tried to ablate CRAF with Cas9 sgRNA, and we found that 35% of the clones, which grew with normal kinetics, nevertheless had all 3 alleles of CRAF knocked out, proving that this is dispensable, at least for proliferation in standard assays. Okay, so thank you.
Thank you, John.